I kept the presentation minimal so that if anyone has observations or questions, please feel free to chime in during the slides. Don't worry about sort of interrupting me. Yeah, so in my email, I think I said I want to welcome feedback about what I'm talking about today, which is really a specific workflow for incorporating newer RNA-seq quantitation methods at Novartis. We previously, just to give a brief overview, the way we're structured, I'm in oncology, which is just one disease area at Novartis. So there are other disease areas, neuroscience, you know what, I can't think of other disease areas because oncology and neuroscience are really big ones. But I think there's probably 25 or so. And everyone's doing RNA-seq because everyone does RNA-seq for their, to drive their biology forward. And so a big use case at Novartis is just looking for gene expression in, you know, drug treatment samples or doing baseline profiling of samples. And so we have a lot of RNA-seq data. So why did we make a new pipeline? When I came to Novartis, everybody was doing RNA-seq, and they were actually referencing my boss, who's Josh Korn. He had this pipeline called Korn-seq, which is really just a customized version of the cufflinks workflow. It's using all standard tools. It's nothing extraordinarily special, but he did adapt the workflow for handling thousands of samples and actually doing genetic fingerprinting and SNP calling from the data. So Korn-seq, right? So everybody was going to my boss saying, you know, we've got these experiments. We want you to analyze the data. The data analysis, just the compute time for the pipeline was like 36 hours. The tools were old in terms of RNA-seq tools. You know, maybe they were 4 or 5 years old, but still that's not fantastically new. And the annotation that we were using was a little bit out of date. So, okay, these alignment-free RNA-seq methods came along. Everybody's sort of more or less familiar with that terminology. They're not really alignment-free. I guess they're like mapping only. So you still place the reads in the transcriptome. As a result, you don't perform a full alignment of the reads. You don't spend as much CPU time on alignment. Everything's faster. That's great for us because we have thousands of samples. And we want to reprocess large data sets, including all of our internal data. But the tooling didn't really feel good that surrounds all these alignment-free methods. As an example, for the cufflinks workflow, there are tools for QCing your data. You're generating alignments. So if you want to see where your reads are mapping and you have some funny looking gene that just seems wrong, and that's really important to your biology, you can go inspect the alignments and you can say, oh, aha, like that's like highly homologous to another gene, and we have to handle it as a special case, that kind of thing. The alignment-free methods don't really have standardized file formats. They don't have tooling surrounding QC, and you actually can't easily fingerprint your samples so that you can identify things like sample swaps, which happen, you know, at a low percentage. But if you have thousands of samples, it's actually pretty significant. And then also, to be honest, our previous pipeline wasn't that reproducible, and it's fantastic that a lot of effort went into it, but at the end of the day, it was just a bunch of bash scripts, and nobody really knew exactly what all the outputs were except the person that created it. And so it placed a huge burden on my boss because everybody would go back and say, what are these files on the file system? I want to reanalyze my data. And it was a huge time sink. Okay. So that's to explain why I created this thing. And it's called PISCES. It doesn't stand for anything, but I'm using salmon as an alignment-free method. So to keep with the sort of fish terminology. The way I think about this is I've created a package that eases the burden of processing a large number of RNA-seq libraries. And as part of this, I wanted to reduce errors in parameter selection. So we have RNA-seq libraries spanning probably not an entire decade, but we have some older RNA-seq libraries generated on older instruments. They might not be paired-end. They might have been generated with a different RNA-seq library preparation than what we're currently using. They might be strand-specific, or they might not be. They might have poly-A selection, or they might not be. We might not even know what species the data are supposed to be, although hopefully we've kept good enough records so that we know they're human or mouse. But in certain cases, we're pulling in data from collaborators, and they haven't kept very good records. So we don't want to trust that these are all human samples. And they might be mouse, or a special case, xenografts, which we have quite a few of. So I break this pipeline down into 3 stages. I think about this as a single sample analysis. So this is one library where, if you can actually read this chart up here, starting with the only thing that we usually have, just the FASTQ files. That's all we need. This pipeline infers parameters for library geometry. It infers the species of the sample, performs SNP fingerprinting if it's a human sample. And then using all of these inferred parameters, we pass the correct set of options to Salmon and quantify the library. So we can do that in approximately 10 minutes per sequencing library. And that's just because Salmon is incredibly fast. We looked at Callisto as well. We liked Salmon for multiple reasons. Ultimately, licensing was one. Callisto, I think, is AGPL, which is sometimes a little bit scary for people in industry. I think in our case, it would have been okay because we weren't making contributions to the code base, and we weren't linking against it as a library. But in any case, Salmon's a great tool. So at the end of our single sample analysis, I have all of these tables of TPM counts data inferred from the reads. Also, a call about a guess at what the organism is, whether it's mouse, human, or xenograft. But I've actually extended this past mouse and human, and we're using this on rat data. We're using it on some microbial RNA-seq data. So this is generalizable. We want to aggregate all of these libraries into one analysis-ready matrix or set of data matrices that you can easily visualize or perform differential expression analysis on. And because we're sometimes pulling in RNA-seq libraries with disparate sample compositions, so we might be comparing a human library against the xenograft library where we have an entire other organism to keep track of. I have a normalization procedure so that comparisons across samples are not problematic in terms of varying sequence content. So you might have one sample that has lots of non-coding RNA contribution, and since TPM is a sort of a zero-sum normalization, you don't actually want those contributions to downweight or affect the protein coding content that you really care about. And then in the same way, we also want to aggregate across all of our QC metrics per library to visualize our QC and flag samples as, pass or fail. So that's a broad overview. The way I developed this.
What's the library?
Yeah, great question. I don't have a slide for that, but just verbally, you can have different geometries where if you have paired-in data, the reads can be facing each other. Usually, that's the geometry that you have. You can have reads that face outward. You could have fragment reads. You can also have, for paired-in libraries, if you have a strand-specific library where you're generating cDNA from from only one of the strands and not generating both the complement and the original strand, you can have the first read start on either the original strand or the complement strand. So you have to actually determine that before you run the quantitation. Actually, since I developed this, I communicated with Rob Patro, who's the Salmon developer, about some of these issues, and he's incorporated library geometry detection into Salmon. So I'm not saying that's completely novel anymore, but at the time, it was a parameter that you had to pass in, and you had no idea what your geometry was unless it was labeled, right? So that really helped us.
What QC metrics are you using?
QC metrics, okay, great. I was hoping that, so at the end of the slides, I have acknowledgements. There was another guy, Jason Merkin, who was working on this with me initially. He came up with this really great idea to use Salmon itself to do some QC. So Salmon ultimately is just counting something like sequence k-mers. And so if you give Salmon a sequence index that contains a downsampled percentage of intronic sequence and ribosomal RNA, mitochondrial sequence, and viral sequence, then you can actually quantify those elements in your sequencing library. And, you know, instead of getting transcript isoform or gene level quantification, you're actually quantifying the things that you don't want in your library, right? So we tried using Salmon itself with a special index built with all these QC elements to generate its own QC metrics. And I think that we could have further developed that approach. Pragmatically, we sort of dumped that early on, and now downsample the FASTQ files to approximately 100,000 reads and perform a bow tie to alignment. That still takes less than 10 minutes per sample, and it gives us enough information to quantify the amount of ribosomal RNA, the amount of intronic sort of genomic contamination, mitochondrial DNA, all of that.
Do you calculate duplication rate?
Duplication rate's problematic if you downsample your number of reads. So I do calculate it. It's not reliable. Usually it has not been an issue, but if we have funny looking libraries and none of the QC metrics that we track explain that funniness, then we can go back and perform a full alignment and calculate the duplication rate.
And most of the libraries are probably made from millions and millions of cells.
Exactly, yeah. We're not using this pipeline for single cell sequencing.
But there are sometimes kind of intermediate level.
Exactly.
But something like tracking number of k-mers, like 20 mers, things like that, first 20 mers in reads, that's kind of gives you a sense for the complexity of your libraries.
Yeah.
Do you have anything like that?
Yes, I do actually. Okay. I have a slide that addresses that.
Right.
The way that I've implemented this for deployment within the company, and hopefully, so I'd like to make this sort of contribution to show other people what we're doing. Maybe some of these methods are useful for other people. This is just a Python 3 package. There's some R code, so I bundled everything together. The R code has dependencies, and I'm managing those using PackRoute, which is kind of like a Python virtual environment for R. So all of this is installable from pip, and all of the parameters are driven by configuration files. Not to say that you have to craft special configuration files for each time you run this pipeline. So I'll talk a little bit more about that, but building transcriptome indexes and supplying sample metadata, defining contrasts. Just a quick overview from December. We have thousands of RNA-seq libraries. This is just internal data at Novartis. We definitely have over 3,000 RNA-seq libraries just in oncology. And I calculated this took something like 30 CPU years on our compute cluster for our previous pipeline. We have like approximately 30 times better efficiency now with this pipeline. really where that matters, we have tons of CPUs at Novartis. So somebody could say, well, your efficiency doesn't really matter because you have lots of compute power, and so just run these on a cluster, distribute all the jobs, and you're done. But really, having a faster, more efficient pipeline allows us to do parameter optimization. We can do things like run all of our RNA-seq libraries through the pipeline, and then we can use the empirical distribution of some of the data and the metrics that come out to drive parameter optimization to make the pipeline even better, and then repeat the process. And we also want to reprocess huge public data sets so that we can bring everything in and with fewer problems have easier comparisons across data sets. So compare our internal data to TCGA and GTEx. The general workflow that I developed, so I want to say with Brad sitting here in the room, there are better ways to make pipelines, right? So I don't want to make a new pipeline framework. This is just driving our use case. So I just want to put that out there. Some of these ideas could be adapted in like a common workflow language pipeline. That's not what this talk is about. You need to make indices for Salmon and for our QC indices, Bowtie2. And I said I have configuration files that drive all of this. Really, the process is just taking in a GTF and a FASTA. For a human mouse xenograft, you end up taking in 2 FASTA assemblies, 2 GTFs, and making a big combined index. The way that I've put this together, you can generalize the process to add extra user-defined FASTA files. That's helpful in our case because I've added viral sequences and consensus repetitive elements. For certain cancer indications, it's actually really neat to see that you have some reactivated repetitive elements. And sometimes viral sequences can make up a pretty large percentage of your transcripts. The way that I've decided to define these index builds is by just JSON that allows you to name a species. This is for a xenograft build. And then you could have multiple indices that you build. So I build a GenCode index, which is just GenCode for human and mouse, passing in the assemblies and GTF files with nothing extra, and then an output location. What I use on a routine basis is GenCode Plus. This is adding in some consensus repetitive elements and some non-redundant viral genomes. And then it's also worth noting that the transcriptome sometimes includes repetitive elements that are expressed in genes. And one big advantage of a method like Salmon is that it does map reads to one read to multiple locations. The functional effect of that in the abundance inference step, it's not clear that you always want to place reads in all possible locations, especially if you're looking at very lowly expressed transcripts and you're trying to quantify things like immune cell infiltrate into a tumor cell. You probably want to have a very low baseline noise in your output files. You'll probably want a reduced sensitivity and higher specificity. So one way I've tried to achieve this reduced sensitivity and higher specificity is to mask anything repetitive in the genome. So the way I'm doing this is just if you have case masking in your assembly, then you can actually hard mask those. And there is a difference in the output. So I'm kind of exploring this idea a little bit further. This is something that should be probably upstreamed into like Salmon or Callisto, and they should probably take care of this in an algorithmic way instead of masking the sequence. Okay, so then the rest of this pipeline is actually just developed so that anybody in Novartis can run it, and they don't have to ask me so many questions, and hopefully it's flexible. So you can pass in any number of read one and read two FASTQ files, or you can just pass in read ones. you can, so that's all you need. Everything else is inferred from the data, the species, the library geometry, and there are sane defaults for like the sample names and that kind of stuff. You can also say this is a xenograph, right? Sometimes we have xenograph samples that have very little mouse contribution. By default, I call something a mouse if it's greater than 95% mouse. Something's human if it's greater than 95% human, and anything in between is a xenograph, right? It's usually reasonable, but sometimes, and that's one case where I've actually run thousands of samples, built an empirical distribution from our real data, and then my observation is that, yeah, the xenographs are usually between 95% human and 95% mouse.
In cases where you have substantial ribosomal RNA contamination, could that impact your apparent species attribution?
No, because I'm not using ribosomal sequence for this. I'm using beta-actin. It's a special case where I'm actually doing my own K-mer counting, just only grabbing beta-actin sequences and then generating a best guess. But yeah, if we had, so beta-actin may not be the best gene for this, and you could extend this using like 16S ribosomal RNA. Then there's a default config file that drives a lot of the default parameters. You can pass in a user config file. This config file is what I have shown for building our indices. So if you have a different set of indices, this is useful for people in a different department who have a different assembly, they have a different transcriptome build they want to use. They can build all of that on their own, keep it in their own user config, and then pass it into the pipeline. So this is just only for our, this is driven by our use cases. And this just has a command line parser. I thought I'd throw it up here because so many sort of pipelining tools that are driven by config files. It's like, well, you craft your config file and then you have this text file and then, oh, it's a make file, right? So everybody knows what a make file is. And that doesn't always work out well for a broad user base. So almost everyone understands what a command line utility is, and you can sort of tweak these options. But like I said earlier, the only required inputs are these FASTQ files. So you run this single sample, one library per sample analysis, and what you get out are sample metrics. I sort of shortened this. I didn't go over this, but we're generating a VCF file. I'm generating a VCF file just based on K-mer counts of 226 high-frequency human polymorphisms. These are also present on the Affymetrix SNP6 array. So we have fingerprinting data for tens of thousands of samples, historic fingerprinting data, and we can match our RNA-seq libraries to the different data that already exist inside our organization by just generating a VCF file from these K-mer counts. I should have put the slide in here because it's actually a great, I think it's a great idea. For every SNP, you just have 4 K-mers. Each K-mer represents top or bottom strand, major or minor allele. And if you just track all of these 4 K-mers for as many SNPs as you need, then you can do a very simple inference of the genotype at that position.
How long are your K-mers?
20.
Okay.
20.
Total, not, so 10 and 10.
Oh yeah, flanking the SNP, so they're 21.
Okay.
So they're 10 bases on each side of the site that you want to genotype. I don't see a lot of people doing K-mer-based genotyping, but I actually think if you have a large amount of data, it's very efficient. So we have all these sample metrics that come out of the pipeline. Some of these sample metrics are fed back into Salmon. You get your TPM matrix and your counts matrix. You can also run, if you provide 10 different transcriptome annotations that you want to quantify per sample, then you can have all 10 of those This supports a use case in Novartis where neuroscience might want to look at oncology data, but they're using in CVI REF-seq annotation, and oncology is using ensemble annotation, and you don't want to go back and re-quantify all your data, so why not just use all of the different transcriptomes that you might care about, and then everybody's happy.
Quick question about analytic assignments. So basically, you're using RNA-seq, you're not using DNA, right, to genotype?
I'm not using DNA to genotype. And that's sort of
my question is about allelic bias in expression.
Yeah, great. There's definitely allelic bias. Typically, it's not bad enough that it affects your call in a way that you have a heterozygote call that becomes homozygous. Sometimes it does. In that case, you may have one SNP out of, in this case, 226 that converts. That's still good enough to match its genotype against another database of sample genotypes and get a high confidence match.
And the question from the other side, do you think it is possible to use k-mers to detect allelic bias? Like, can you try it on imprinted genes?
From RNA-seq data, I think you would have to have the DNA-seq as well. Because to say there's allelic bias, you have to know that there are alleles. Yeah. Yeah, it's problematic to do SNP calling from RNA-seq data. That being said, we do use SNP calls on a routine basis, and the previous cufflink space pipeline that we have, we still use that for SNP calling, genome-wide, right, for every sample. And that's always a concern, that you have allelic bias. Yeah. Just really quickly, okay, what do the outputs look like? There was a question earlier on about overrepresented sequences in these libraries. So I take care of that. Some of the sample metrics are basically FastQC. But I, as an exercise, I cloned it in Python, and the reason this is nice is that my FastQP package, I can customize the output, the figures that we produce, the tables that we produce are all tidy data. And one of the biggest pain points for me is FastQC produces this text output that's like a table that a human can read, but it's not even really good for humans reading it, and it's terrible for machines reading it. So all the sample metrics from FastQP are just a tidy table where each observation has a variable and a value, and you can just read that into your database very easily. We have this VCF file, which, as I just described, is generated from k-mer counts, and I use this to fingerprint the samples. So every time this pipeline runs, I already have built a database of all of our baseline profiles, genetic profiles for all the samples that we know of at Novartis. And so every time I run a new sample through, I get its VCF file, but I also get a label so that the highest confidence match of this sample is the cell line that I think it is, or it's not. And I haven't quantified it, but our rate of sample mix-ups is, less than 10%, way less than 10%, but it's more than like 1%. So it adds up, and it really matters. And then these downsampled alignments, just doing full alignments with Bowtie 2, generating some QC metrics, and then aggregating these QC metrics. So now, just another command line parser where you don't have to provide really anything other than the paths to the data directories. And this gathers all your statistics into a table. It's tidy. This also takes all of the VCF files in an experiment and fingerprints them against each other, which you can use to then generate an identity matrix where you have probabilities of sample matches, and we can identify sample swaps within an experiment. I didn't put a picture of this. I should have. it's just a clustered heat map, right? So if you have 4 cell lines in an experiment, you should expect to have 4 clusters. And if you have 5 clusters, then you have a big problem. So this saves a lot of time up front for us. One pain point for me is that Novartis really likes this Spotfire tool. I had never heard of it before I started the job. And some of you might know about it, some of you might not. For better or for worse, it's what everyone at the company uses. It's actually a great interactive visualization tool, but it's prohibitively expensive, and it's also not very flexible. That being said, I tailored the tidy output from this summarization tool to be read into a template where we can generate these plots. Just briefly, I've highlighted a sample where we have more energetic sequence than we would expect compared to all of the other samples in the experiment. These are all a universal human reference RNA sample. So these are control samples that are run through our sequencing facility in conjunction with every experimental sample that comes in. So we've generated a couple hundred of these UHR samples, and so they should all be identical. This one's not for some reason. Probably a sample swap, or this is actually probably a library prep issue. This looks like a library prep issue. You can also see, so this is fraction coding, energetic, intronic, UTR. You also get an estimate of the amount of autosomal RNA, mitochondrial, ribosomal, and viral. And then a normalized view across all genes of the 5' 3' bias. Again, we tried doing this by splitting up all of the genes, all of the transcripts in the GTF just into their 3' and 5' portions, and then using Saman to quantify just the 3' end and the 5' end of all transcripts. And then we could have some rudimentary measure of the ratio of 5' to 3' the sequence content. That worked pretty well, but at the end of the day, just downsampling and running the existing tools took the same amount of time, and it's proven. So to answer your question about duplication rates, that's also in here somewhere too. Some of you are probably familiar. These are just Picard QC statistics. Let's not spend a lot of time on this. It's important though. I mean, so the point I want to make is the pipeline gives you QC data. It gives you QC data in a tidy matrix that you can visualize. We're doing visualization using Spotfire. You could actually just read it using your favorite tool. Okay, so then you also want to summarize all of your expression data into a tidy matrix. The way that I've laid this out, you can just pass in all of the sample directories where you have your analysis files. The pipeline will then do the right thing and put together abundance matrices. Everything's labeled by directory name. It's not very nice, but if you just want to get all of your data together and have a quick look, it's fast. You can also pass in a metadata file, which gives you sample identities, treatment variables, cell line names, anything that you want to label your data with. For visualization purposes, Spotfire is a great tool for visualization, but it can't really do great calculations on your data. So I also output a log fold change matrix, which is normalized by variables defined in this metadata file, grouped by, normalized by treatment, and the control factor is DMSO in this sample. So that just gives you log fold change values across your data matrix for making a heat map. That's all this is. Then since I'm trying to process tons of RNA sequencing data, and I'm not the only person in our group that does the RNA-seq analysis, but I'm pretty much the only person in our group that does the RNA-seq analysis. I meet with every person that's submitting a new experiment in the oncology department and go over their experimental design, provide guidance on updating their experimental design, ask, what's the question that you want to answer? Will this actually answer that question? I do power calculations. And then I work with the sequencing facility to actually get the sequencing data pushed through, and I coordinate all the communication. the end of the day, I actually have to do the analysis. And usually that's just doing differential expression between treatment and control groups. Because that's a huge use case, I define my contrasts for DE-seq in a YAML file, and I pass in a PATSI formula that's logged, so this is all reproducible. For me, that's important because 5 months later, if somebody asks me to go back and look at what I did in their experiment, I have no idea. And this also allows other analysts in the group to go back and see, okay, well, this was the analysis. It's not just some R script that nobody documented, right? You actually have labels. Okay, so to briefly summarize here, you get tables of all your genes. So isoforms, genes are summarized from the isoform level data using, these are all open source tools. This great TX import function that's now in the DEC2 package, Mike Love developed. And this handles all the file input. And Stephen Turner has this nice dump of Biomart, which allows you to annotate genes with all kinds of extra data, in this case, like gene symbol and entree ID and ensemble ID and that kind of stuff. And then all the outputs are tidy. And one last point that I want to make, and I don't have a slide about this. It's tough for me to put together a slide because this is a continuous problem and I don't have a fantastic solution for it. But normalization across samples, it's extraordinarily important. I think earlier I mentioned that you might have one sample that has more non-coding content than another sample. Using the GenCode transcriptome annotation, and that includes roughly 60,000 genes, many more isoforms, but only about 20,000 of those genes are protein coding. Sometimes a sample will have just one or two non-coding genes that are highly abundant. TPM abundance estimates then are entirely miscalibrated for the rest of the protein coding subset of the genes that you care about. So what I've found, and this is just sort of empirical, I've run all of our data through the pipeline, identified samples that look terrible, determined that there are certain subsets of genes that are problematic in that sample. So mitochondrial and ribosomal genes, so not ribosomal RNA, but like there are genes that encode the ribosomal proteins. Those are problematic. So I exclude those from any normalization calculations. Mitochondrial genes, those also tend to be problematic. For certain indications, you have lots and lots of non-coding content, like I would keep mentioning. So it's just great if you don't care about the non-coding genes most of the time, you can just exclude them from your normalization calculations. And I'm using TMM, which is trim median of M values. And this is a function that's in the EdgeR package to calculate scaling factors. Okay, so I only put the protein coding genes into this normalization procedure, come up with a scaling factor, and then I apply the scaling factor to all classes of genes. So what I end up doing is I make the protein coding subset of the genome, transcriptome, more comparable between samples. Sometimes inflate the non-coding portion, which is problematic if you care about the non-coding elements. And if we're sequencing from, say, PBMCs or whole blood, you end up having hemolysis in your samples sometimes. So then if I have those sample types, I will also pass in a list of genes like hemoglobin that are sometimes highly abundant in those specific samples and exclude those from normalization. So normalization is not easy, and it's not the same for every sample. In addition, xenograft samples the way I handle xenographs is that I build a transcriptome index that includes all human and mouse genes. Well, Salmon quantifies all of the genes, but then you don't really have TPM. You have TPM of so transcripts per million for both human and mouse transcriptomes combined. And then if you want to compare only the human portion to another sample that's only, it's the same cell line, but it's grown as a cell line, not a xenograft, you have to rescale all of the human genes from the xenograft back up to 1 million. And then normalize all the samples together. Okay, so I don't have a slide about this. It's an area where I'm actively tweaking parameters and it's still a concern. And if we have time, I'd love to hear anybody's ideas about how to normalize multiple samples so that your abundances are more comparable.
Two quick questions.
Yeah.
About mitochondrial stuff. The first is you're talking about mitochondrial genes or mitochondrial proteins that are coded in nuclear.
In this case, both. Yeah, so mitochondrial genes are probably more problematic because you have multiple copies of the mitochondrion. But mitochondrial proteins encoded in the nuclear genome also tend to look funny. I don't really have data to back that up, but they just do look funny.
I'm asking because this might be important observation. People have been reporting a lot of mitochondrial changes in acid, right?
Of course, yeah.
And whether it is artifact, I'm not sure now.
Yeah, so I think it could be an artifact. I think it's important to understand the question that you're trying to answer with your sequencing data, right? So most of the time the question is, what are the differential changes in the protein coding subset of the transcriptome? If you normalize your data so that you can answer that question, it doesn't preclude you from answering questions about the non-coding portion or the mitochondrial portion. It just means that you have to make sure that normalization makes sense.
But you say that mitochondrial stuff is funny. It can be basically, the difference can be due to this funniness.
The difference could be due to this funniness. I think that it's actually probably real. Because, and the reason that I say that is, so the sequencing library that you submit and then sequence, the data that you get back, they're not normalized in any way. So whatever goes into the sequencing library is what you get out. And for certain ribosomal RNA, right, you know where that comes from. And you know that you've tried to remove that, hopefully, in your library preparation. The mitochondrial genes that are encoded on the mitochondrial chromosome, that, you know where that came from. But anything that's autosomal, that's highly abundant in your sample before you do any normalization, that's probably real, right? So then the question is, if you want to do differential comparisons on the subsets, say like mitochondrial genes that are on the autosomes, then how do you normalize your data? There's a real difference. Yeah, it's biologically real. And I would say probably you don't do any normalization if you're just looking at a specific subset of genes like mitochondrial autosomal genes. Unless you have contamination from non-coding genes in the same samples. So it depends highly on the sample makeup. And that's why I don't have a very good answer for a standard normalization procedure.
So mitochondrial thing is not a good factor right now.
Yeah, it's just a trade-off that I'm making, right? So again, yeah, if anybody has ideas about what normalization is proper.
I mean, extending your TMM to scale all genes, I'm a little disappointed by that. But
so I think, right, after you've TMM normalized your values that were originally TPM, they're not TPM anymore. So they won't sum to any value. They're unbounded. When I say I'm TMM scaling the protein coding genes, I'm really only coming up with scaling factors using the protein coding genes. Those scaling factors, so you're disquieted that I apply those scaling factors to all genes?
Well, you remove autos. Are you applying them back to all the ones that were moved in the?
Yes. Yes, sorry, that's what I was trying to communicate.
Well, you remove, how can you make assumptions about the space of, the proportion of reads and then the transcriptome mass occupied by those that you remove before you calculating your scaling factors?
One thing that I forgot to mention here. So you're worried about transcriptome mass, right? Okay.
Yeah,
so you're worried that the scaling factors will actually, you're worried that if you have huge contributions by sort of non-coding genes and you exclude those from normalization factor calculations, that you'll be overestimating whichever one. What I forgot to mention here is that to make this normalization scheme appropriate, I actually rescale the TPM data before I do any TMM normalization by taking all the protein coding genes, summing their values, and then I scale those all up to 1 million. So that you start with TPM of just the protein coding subset of the transcriptome, and then you calculate scaling factors just on the protein coding genome. So effectively what you're doing is you're saying, okay, I have this gen code annotation. Let's throw out all the 40,000 things that we didn't care about. Pretend like we ran our quantification pipeline using only 20,000 genes to get TPM. Then let's scale that appropriately. And then all of this other stuff that we've effectively removed, let's also scale that too.
Why? That's final step.
Yeah, I don't know that it's appropriate. I haven't had any experiments that hinge on the success of a differential analysis of the non-coding portion of the transcriptome.
Why going back to them if you exclude them, if you don't want to look at them? I think you don't need to do that. I'm a bit worried about applying those factors.
Yeah,
we can keep.
Sure, I know this is the feedback that I was hoping to get actually. Yeah, I think it's problematic, and maybe what I'm doing is not appropriate. I'm definitely willing to admit that.
Although, I think your way could extend. What if you're comparing a sample that's total RNA versus poly A or RNA, and you want to minimize the effect of the sample preparation? Then I would think you could essentially get your scaling factor from poly A transcripts and then apply it to everything. Right?
Yeah. So thanks for saying that, because that's actually one of the use cases that drove me to come up with this normalization procedure. We have a mixed number of samples with different library preps. And so it's very easy just to look only at the poly A subset of the genome, right? And then say, okay, that's what we care about. Let's get it right. And let's figure out the rest of it later.
And I'm perfectly fine with that. It's just the idea of going back to non-coding that wasn't part of the normalization.
Right.
And as long as we start certain coding, I'm perfectly fine with it. It's actually very close to what I'm doing.
Yeah. Okay. That's good to hear. So just in the few remaining slides, I wanted to describe, I think the implementation is sometimes important. This is what works for me, and this is what's working for us as a group. So just CSV files, right? Just encoding all of your treatment information, sample IDs. We have universal identifiers for all of our samples of Novartis, so pulling those in. And then any other treatment variables that you might care about. So the format for these metadata files is just, at a minimum, a sample ID column. If you pass in a directory column, it also then allows you to drop passing in directories to the command line interface. You can then pull out your analysis directory from a column in your metadata file, which is sometimes helpful if you have 100 samples or hundreds of samples. You don't want to pass a command line that has hundreds of directories in, but that's easy to mess up. The metadata is actually used then in the differential expression analysis, where you can then define the treatment variables that are in this metadata file in a YAML format, where you set up your experimental variable and then the base level of the variable that you want to normalize against, and a formula for setting up your model matrix. And then you get out just a tidy table of your differential expression p-values and fold changes. Defining the contrasts in this way has also been useful because we have, as a big company, databases where we want to ingest and integrate all of our internal data. And if we have p-values and fold changes, you want to know how those p-values and fold changes were calculated and which contrasts were pulled together. So this allows me to construct all of that, metadata about the analysis. Again, I have a command line parser for this. You can sort of tweak individual options, but everything has a default, and you only have to pass in directories if that's all you have. Sorry for all the screenshots in here. I didn't know what to do with this because I'm talking about I'm making text matrices, so that's not the most interesting thing to present. But I just want to point out, at the end of this, you'll have analysis ready counts, a tidy table of your p-values and fold changes, tables of TMM scaled log fold changes and log fold changes without the scaling. So I always make sure that I keep the raw data as well as the normalized data, as well as the TPM data. And then some extra annotation. This is where I'm using the notables package to pull in things like gene symbol, and this is actually very important for my normalization procedure, which is the biotype. So protein coding is typically all that I'm comparing against. Our visualizations are pretty standard. Again, Spotfire is a blessing and a curse, but having this pipeline allows me to go from unprocessed individual RNA-seq libraries to a summary matrix and visualization in these templates that I've created in less than an afternoon, like maybe 2 hours, including the hands-on time for loading everything into this template. So all of what I'm presenting is really driven by my use cases, and my biggest use case is speed, right? So I want to get the turnaround time from talking with the biology groups to sequencing to analysis down as quickly as possible. And sequencing, honestly, is the thing that takes the longest, but everybody's frustrated when they've submitted their samples for RNA-seq analysis and the sequencing has been done and the analyst just hasn't communicated anything back. And, I'm trying to make that happen less and less. I don't know if that's a problem for you guys, but I think there's a big bottleneck for the bioinformatics analysis. Okay, so to summarize, I have some near-term development goals. I want to focus a really big portion of my time on normalization efforts with our internal data sets, but I think that there are some big takeaways for normalization using these GenCode and just generally expanded annotations that include a lot of sequence elements that we don't properly understand. There are some new tools for borrowing information about abundance, posterior probabilities across samples. Rob Patro's developed this tool called Shoal that I haven't really evaluated yet, but I think that's something that's on my radar. I have this automated re-identification of samples against a multi-sample VCF. If I distribute PISCES publicly, I'd like to have a pre-built database of common samples that you might encounter in public data sets and common cell lines. Novartis has the comprehensive cell line encyclopedia, which is about 1,000 cancer cell lines. NCI has the NCI, whatever, is it NCI 60 something? Not that many anymore. So I'd like to actually distribute a database that can be used for sample re-identification. I think that's important. And open source visualizations. So if anybody has great ideas about a way to replace Spotfire in my workflow, and I'm open to anything up to and including something like a Shiny server instance, but what I'm envisioning is if I distribute a pipeline that packages a bunch of tools and produces analysis and visualization ready tables, I don't want somebody to have to spin up an R server and set up Shiny and load all of their data into, sort of a browser visualization tool. So I don't think something like this exists, but what I'm envisioning is, something interactive, something that maybe runs on your browser, spawns a local web server from your computer, and allows you to make nice volcano plots and heat maps, that kind of stuff. All I see right now is that I need to go make my own D3 visualizations, and that's not really in my job description right now. I don't have time to put together a visualization package. But I see that the general community does not have very good tools for visualizing RNA-seq data that are open source and easy for a single analyst to run. It doesn't require a lot of infrastructure.
Multi-QC is pretty good. We use that. I mean, you have to generate plots. But,
so multi-QC is great for the QC data. It's not
really interactive, but sort of.
Yeah, and it's not really meant right for visualizing the abundance data.
Could do.
Could do.
Just
Okay.
plots, right?
Yeah, I don't know how.
Yeah.
Maximum of time.
Yeah.
I remember something like 200 was okay. I don't know.
Okay. I'd love to explore that more. I've actually branched multi-QC and made the changes necessary to incorporate my QC tool, FastQP, into multi-QC, just because I thought that would be fun. And so, who's the primary developer for that?
Phil. Google. Yeah. Stop.
I just don't know.
Yeah, Sci-Life Lab.
Okay.
Yeah, he's cool. He's done very fun.
Okay. Yeah. So for publication, I want to provide visualizations. I think it's a disservice to stop it. Just like here's a data matrix. All right. So maybe I convince you that I developed this to solve some real world issues. This is not a general purpose. This is the pipelining framework that you should be using, kind of thing. But Pisces has been really valuable for our department. Like I was saying, my job is to handle all aspects of RNA-seq from the biology groups in our oncology department. So tool development's not really my main task. It's not my job description, but in this case, I did develop something that we're using as a tool internally, and I think since it's built on mostly open source tools, we're going to try and publish this as a preprint at least. I don't know exactly the utility of having a preprint that describes our pipeline. I have documentation right now, but I think the general idea that I have is I want to make this available so that the ideas can be inspected, I guess, and I can have a conversation around best practices or tooling that we can develop to enhance the utility of these alignment-free methods, because they're really cool, right? And it makes my job a heck of a lot easier. Everything goes from like a week of processing time to an afternoon, and that just means that I get to spend more time looking at the data. So anyway, my colleagues, my boss, Josh Korn, I didn't put Vivek's last name, that's a mistake. Not that you would know any of these people because they're all NIBR, but sorry, yeah, Jason Merkin, he was an MIT grad student, and he's at KSQ Therapeutics now. So yeah, so maybe he'll present something here next year. I don't know what he's doing. And then Rob Patrick. So he's been really great to work with. He's a very responsive developer. I think he's a fantastic example of somebody in the open source community that is close to the science and knows both the science and the computer science. So. And yeah, I'd love to have any kind of conversation. Online conversations are very welcome. I threw my handle up here. I'm MD Shaw 5. That's just a remnant from my college days. It was a random email username, but I am that on Twitter and GitHub and pretty much everywhere else online. So.
Can you send those slides around to us?
And I'm definitely going to send the slides around. And if Brad points me to the video recording, I'm going to post that as soon as possible on my website because I think that's pretty cool.
Cool. Yeah, I'll upload to YouTube and then I can put it to the big thing.
Fantastic. Cool. I really appreciate you guys being here, and I haven't been as involved in these meetings as I would like to be, but I really like the idea. So, so thanks for organizing these. I guess I don't know who I'm thinking.